Ever wondered how DNA sequencing works? Well, today we delve into the fascinating world of the chain termination method. DNA sequencing is a crucial scientific technique that allows us to unravel the intricate genetic code embedded within every living organism. This genetic blueprint holds the keys to understanding life itself, from hereditary traits to disease susceptibility and much more. Among the various methods available for decoding this complex code, one stands out for its efficiency and simplicity. The chain termination method, also known as the Sanger Dideoxy Method, it has been a game changer in the field of genomics. Using fewer toxic chemicals and less radioactivity than other methods, it's an approach that has greatly simplified the process of DNA sequencing. By the end of this video, you will have a solid understanding of the chain termination method and how it has revolutionized DNA sequencing. So, how does the chain termination method work? Let's break it down. This method is a brainchild of Frederick Sanger, and it primarily hinges on the use of dideoxynucleotide triphosphates, or DDNTPs, as DNA chain terminators. To understand this, imagine the DNA as a long chain of building blocks, and these DDNTPs are special blocks that halt the construction midway. This interruption gives us a series of DNA fragments of varying lengths, which are instrumental in deciphering the DNA's sequence. Now, let's talk about what we need to get this process started. First, we need a single-stranded DNA template. This is the DNA strand we're aiming to sequence. Next, we need a DNA primer, a short strand of nucleotides that binds to the DNA template and kickstarts the process of DNA synthesis. We also need a DNA polymerase, the enzyme that facilitates the addition of nucleotides to the growing DNA strand. Then we have the nucleotides, the building blocks of DNA. These are labeled either radioactively or fluorescently, which helps us track their incorporation into the DNA strand. Finally, we have the stars of the show, the modified nucleotides, also known as the dideoxynucleotides. These are the chain terminators, lacking a crucial component that prevents them from forming a bond with the next nucleotide, thus stopping the DNA strand from growing further. The DNA sample is divided into 4 separate sequencing reactions, each containing all 4 standard deoxynucleotides and the DNA polymerase. To each reaction, only one of the 4 dideoxynucleotides is added. When the DNA polymerase incorporates a dideoxynucleotide into the growing DNA strand, it causes the DNA synthesis to stop, resulting in DNA fragments of varying lengths. The position of each fragment within the sequence provides a clue to the sequence of the original DNA strand. With these materials in place, the stage is set for the sequencing process to begin. Now that we have everything we need, let's get sequencing. To begin the sequencing process, our DNA sample is divided into 4 separate reactions. Each of these reactions contains all 4 of the standard deoxynucleotides, which are crucial building blocks of DNA, along with the DNA polymerase, an enzyme that plays a key role in the replication of DNA. Also added to each reaction is one of the 4 dideoxynucleotides, special nucleotides that are the true stars of the chain termination method. These dideoxynucleotides are what we call chain terminators. Unlike standard nucleotides, they lack a crucial component, the 3O group. This missing group makes it impossible for the formation of a phosphodiester bond between 2 nucleotides, thus halting the extension of the DNA strand. What we end up with is a collection of DNA fragments, each of different lengths. But why different lengths, you may ask? Well, the incorporation of the dideoxynucleotides into the DNA strand is a random process. Sometimes a chain terminator might get incorporated early on in the sequence, and other times it might get incorporated later. This randomness results in fragments of varying length, each ending with the dideoxynucleotide that was added to the reaction. The next step in the process is to separate these fragments by their size. This is done through a process called gel electrophoresis. The fragments are heated to separate the strands and then loaded onto a gel. When an electric current is applied, the fragments move through the gel, with smaller ones moving faster and farther than larger ones. The end result is a sort of DNA ladder, with each rung representing a DNA fragment of a specific length. These fragments, each representing a sequence of DNA, are now ready to be analyzed. But how do we read these fragments? And what can they tell us about the original DNA sequence? All that and more will be covered in the next part of this series. So, stay with us as we dive deeper into the fascinating world of DNA sequencing. So, we have our DNA fragments. What next? Well, it's time for some heat. These DNA fragments are heat denatured. This process disrupts the hydrogen bonds between the bases, causing the two strands of DNA to separate. Now we have single-stranded DNA molecules, which are ready for the next step. The separated DNA fragments are then run through a process called gel electrophoresis. This procedure sorts the fragments by size, with the smallest fragments moving the fastest and furthest along the gel. As the fragments move, a dark band appears in a lane. This band indicates a DNA fragment that resulted from chain termination. Each band represents a different size fragment, and the location of the band tells us the final nucleotide of that fragment. By looking at the relative position of the different bands among the 4 lanes, scientists can read the DNA sequence. They start from the bottom and read upwards like a book. This process allows scientists to read the DNA sequence letter by letter. So what are the key takeaways about the chain termination method? Firstly, it's important to remember that this method is highly efficient and uses fewer toxic chemicals than other DNA sequencing methodologies. It's centered around the use of dideoxy nucleotides to terminate DNA synthesis, yielding a series of DNA fragments whose sizes can be measured by electrophoresis. This process occurs in 4 separate tubes, each containing a different dideoxy nucleotide and all including a radioactive DATP, ensuring the DNA products will be radioactive. However, it's not without its limitations, such as the non-specific binding of the primer to the DNA, which can affect the accurate readout of the DNA sequence. Additionally, DNA secondary structures may also affect the fidelity of the sequence. Technical variations of chain termination sequencing include tagging with nucleotides containing radioactive phosphorus for labeling or using a primer labeled at the 5' end with a fluorescent dye, which facilitates reading in an optical system for faster and more economical analysis and automation. With its pros and cons, the chain termination method remains a critical tool in the field of DNA sequencing.